Welcome back, dear students. In the previous classes, we learned about the digestive system. That means the organs which are involved in the system and their anatomy. In this class, we are going to learn about the different kinds of glands which are involved and their action. Okay, let us start. This is the introduction about what we are going to learn in this class. We are going to learn about the physiology of digestion, digestive glands, and about the enzymes. In the physiology of digestion, there are 5 steps mainly. They are ingestion, digestion, absorption, assimilation, and digestion. Ingestion means nothing but intake of the food, and digestion is the actual process. Okay, here we learn about the different kinds of enzymes which are involved and their action. So food is physically and mechanically masticated, and chemically it is broken down. Okay, so their complex bonds are broken down and they convert into simple soluble substances. Okay, next we learn about the absorption. Absorption is done in the small intestine. Here, whatever the broken down simple soluble substances or food molecules are there, they get absorbed and enter into the bloodstream. The water that we take is also get absorbed in the small and large intestine. The next part is assimilation. This is nothing but the passage of the nutrients or food molecules at the cellular level. Okay, so whatever the food absorbed reaches to each and every cell so that it can carry out respiration process. In the cellular respiration, the energy is released with which we are able to do all these activities. Okay, next digestion. It is nothing but defecation. That means the whatever undigested waste which is created out of digestion will be eliminated. Okay, so this is digestion. And we learn about the digestive glands as well. There are different kinds. There are 3 pairs of digestive salivary glands in the human body, and we learn about the liver, pancreas, gastric glands which are present in the stomach, intestinal glands which are present in the small and large intestine. And we are going to learn about the different enzymes which are existing in the secretion of all these glands. What is actually an enzyme? Enzymes are chemical substances released by glands which acts as a catalyst in the biochemical reaction. That means they help in the process of action. Okay, so they are actually, these catalysts help in breaking down complex food substances into simple soluble substances. Okay, let's go to the next slide. First of all, we learn about the salivary glands. Salivary glands are of 4 types. They are parotid glands, sublingual glands, submaxillary glands, and infraorbital glands. Let us locate their location in the human being. The first gland and the largest one is called parotid gland. It is present below the ear. The location is below the ear. I am locating it here. This is the first one, salivary parotid gland. The next one is sublingual. The name itself says that it is present below the tongue. Okay, so here is the sublingual salivary gland. The third one is submaxillary. Maxilla ante davada. సో దవడ కింది భాగంలో ఉంటుంది కాబట్టి దీన్ని సబ్ మ్యాక్సిలరీ సలైవరీ గ్లాండ్ అని అంటాం. వీటిలో ద లార్జెస్ట్ వన్ ఈస్ పెరూటిడ్, ద సెకండ్ లార్జెస్ట్ వన్ ఈస్ సబ్లింగువల్ గ్లాండ్ అండ్ ద స్మాలర్ వన్ ఈస్ సబ్ మ్యాక్సిలరీ. 4వ రకం ఏంటంటే infraorbital salivary glands. These infraorbital salivary glands are absent in the primates. Primates ante monkeys, chimpanzees, human beings, gorillas, ఇవన్నీ ప్రైమేట్ గ్రూప్కి చెందినవి. సో ఈ యానిమల్స్లో అంటే మ్యామల్స్ లోపల ప్రైమేట్స్లో మాత్రం ఈ ఇన్ఫ్రా ఆర్బిటాల్ సలైవరీ గ్లాండ్స్ అనేవి ఉండవు. సో ద హ్యూమన్ బీయింగ్ contain only 3 types of salivary glands. They are parotid, sublingual and submaxillary. ఈ infraorbital salivary glands అనేవి they are existing below the orbits. Orbits అంటే ఐబాల్స్ యొక్క సాకెట్ ని మనము ఆర్బిట్ అని అంటాం. సో దే ఆర్ ఎగ్జిస్టింగ్ బిహైండ్ ది ఆర్బిట్స్. ఓకే, సో దీస్ ఆర్ infraorbital salivary glands which are absent in the human beings. And composition of saliva లోపల యాక్చువల్గా ఏముంటాయంటే వాటర్ ఉంటుంది, మ్యూసిన్ ఉంటుంది, ఎలక్ట్రోలైట్స్ ఉంటాయి, ఎలక్ట్రోలైట్స్ ఆఫ్ సోడియం, పొటాషియం, క్లోరిన్ అండ్ బైకార్బోనేట్. సలైవరీ అమైలేజ్ అనే ఎంజైమ్ ఉంటుంది అండ్ లైసోజైమ్ అనే ఇంకొక ఎంజైమ్ ఉంటుంది. This salivary amylase is also called as ptyalin. Okay, ptyalin అని కూడా అంటాం దీన్ని. And it is also called as alpha amylase. Salivary amylase అన్నా, alpha amylase అన్నా, ptyalin అన్నా కూడా ఒకటే. So the action of salivary amylase is it converts the carbohydrate or starch into dextrine and maltose. స్టార్చ్ అనేది కాంప్లెక్స్ కార్బోహైడ్రేట్ లేదా పాలీసాకరైడ్ అని అంటారు దీన్ని. ఇది డైసాకరైడ్స్గా కన్వర్ట్ అవుతుంది. ఓకేనా, సో స్టార్చ్ ఈస్ బ్రోకెన్ డౌన్ అండ్ కన్వర్ట్స్ ఇంటూ డెక్స్ట్రైన్ అండ్ మాల్టోస్. దీస్ ఆర్ డైసాకరైడ్స్ అండ్ దిస్ ఈస్ డన్ బై విత్ ద హెల్ప్ ఆఫ్ సలైవరీ అమైలేస్. లైసోజైమ్ అనే ఎంజైమ్ ఇట్ ఈస్ యూస్ఫుల్ టు కిల్ ద బ్యాక్టీరియా ప్రజెంట్ ఇన్ the food that we take. The pH of the saliva is 6.8. 6.8. Remember, మనం ఒకవేళ యాసిడిక్ ఫుడ్ తీసుకుంటే so the food becomes the saliva becomes slightly acidic, and if we take the alkaline food it becomes slightly alkaline, but the pH is 6.8. It is asked many times in the NEET exam. So let us learn about the next slide. Let's learn about the gastric glands. Gastric glands are located in the stomach. As we learned earlier, there are 3 parts in the stomach. The upper part is called cardiac stomach, middle part is called fundic stomach, and the last part is called pyloric. In the cardiac stomach we find cardiac glands, and they are important in secreting the mucus. Mucus is a slippery substance. And the second part we find fundic and oxyntic glands. There are different cells in this fundic stomach: neck cells, peptic or chief cells, parietal or oxyntic cells. Remember these 3 types of cells: peptic, neck cells and oxyntic cells. Neck cells secrete the mucus, which is the slippery substance. Chief cells or peptic cells secrete pepsinogen and prorenin. It is double N actually, printed wrong. Parietal or oxyntic cells, they secrete HCl and CIF, means calcium incentric factor. Okay, and the next part is pyloric stomach. It has neck cells and endocrine cells. Neck cells secrete mucus, endocrine cells secrete the gastrin, which is actually a hormone. Okay, we will learn about the function of this pepsinogen, prorenin, HCl and CAF in the physiology of digestion, that is in the next slide. Let's learn about the digestion in stomach. In the stomach we find 2 kinds of digestion: mechanical digestion and chemical digestion. Mechanical digestion is nothing but the muscular contractions created by the walls of the stomach. We learnt in the earlier class that walls of stomach contain 3 kinds of muscles. They are circular muscles, longitudinal muscles and oblique muscles. These muscles create peristaltic movements which helps the food to mix with the gastric juice. In gastric juice we find HCl, pepsin, renin and CIF. Let's learn about the HCl. HCl is a very strong acid released by the oxyntic cells of the walls of stomach. Its main function is to kill the pathogens. So whatever bacteria, fungus and all, whatever the pathogens are present in the food, they are completely killed by this HCl. And HCl also provides an acidic condition for the functioning of proteolytic enzymes. So these proteolytic enzymes acts on the proteins only in the acidic condition. So HCl helps in providing such a condition. HCl activates the pepsinogen and prorenin. This pepsinogen and prorenin are inactive forms. They are converted into pepsin and renin, which are active forms. Pepsin and renin acts upon the proteins and they will converts them into simple substances. So auto stimulation is done by pepsin and renin. So once the HCl activates this pepsinogen and prorenin into pepsin and renin, these active pepsin and renin enzymes can activate pepsinogen into pepsin. That means they can activate themselves. Hence they are called auto stimulating substances. The process is known as auto stimulation. What these pepsin and renin do? Pepsin converts proteins into proteases and peptones. Okay, so proteins are converted into proteases and peptones, and renin converts casein into calcium paracasinate, and in turn proteases and peptones. Okay, so renin important to convert casein, which is a protein, into calcium paracasinate, and that is further divided into proteases. Renin is found in the children. Okay, in the infant stage mostly this renin is found. So gastrin, we learn about the hormone gastrin in the further slide. CIF, which is Castle intrinsic factor, it helps in the absorption of vitamin B12. Okay, so we learnt about the HCl, pepsin and renin and CIF. I will brief you. HCl is mostly important for killing the pathogen and they provide acidic medium, and they activate pepsinogen and prorenin into active forms called pepsin and renin. Next, pepsin and renin. Pepsin converts proteins into proteases and peptones, and renin converts casein, the protein called casein, into calcium paracasinate, and in turn proteases and peptones. Let's learn further. Let's learn about the digestion in the small intestine. The partially digested acidic food, which is called chyme, enters into the small intestine. So whatever the food enters into the esophagus is called a bolus, and the food enters into the small intestine is called as chyme. So entry of chyme stimulates the walls of intestine to secrete two hormones. They are secretin hormone and cholecystokinin hormone. Secretin hormone stimulates the pancreas to secrete pancreatic juice, and cholecystokinin stimulate the gallbladder to produce the bile juice. To release the bile juice. Bile juice is produced by the liver and stored in the gallbladder. All the food molecules are completely digested here, and it is the site of absorption. Okay, so we learn about the liver and pancreas in the next slide. Let's see the structure of liver. We already learnt about the liver. Liver is the largest gland and largest internal organ in the body, And liver basically has two lobes, first lobe and second lobe. There are two lobes, and if we observe the TS, transverse section of the liver, we find several lobules inside. So these several lobules are called hepatic lobules. Okay, it is the largest gland located in the abdominal cavity and contain two lobes. Each lobe is made of several hepatic lobules. Here you are able to find the hepatic lobules. It is the structural and functional unit of the liver. So the hepatic lobule is the site where the actual process takes place. Lobes are widely supplied with the blood vessels. Here you are able to see the veins and arteries are very widely supplied in the hepatic lobule, and the layer around the each lobule is called as Glissen's capsule. Okay, so each hepatic lobule is covered by the connective sheath. Okay, this connective sheath is called as Glissen's capsule. Hepatic cells, you are able to see here. These cells are called hepatic cells. They make up the cord-like parts. You are able to see different cords, and these are called hepatic cords. They are actually made by hepatic cells. They secrete the bile which flow through the bile canaliculi. Okay, so these hepatic cells secrete the bile juice, and which is carried by the hepatic canaliculi. Okay, so the bile enters into the canaliculi, and then they enter into the hepatic duct and goes to the gallbladder and store there, and from the gallbladder it released by the cystic duct, and cystic duct joins with the pancreatic duct and becomes a common bile duct, and it enters into the ampulla of Vater and finally into the duodenum. Okay, so you must remember that hepatic lobe is made of several hepatic lobules, and the hepatic lobule is a structural and functional unit of the liver, and each hepatic lobule is made of hepatic cords, and hepatic cords contain hepatic cells, and these hepatic cells secrete the bile juice. Okay, let's learn further. Here is the pathway of bile secretion. Mainly they are secreted by the hepatic cells of the hepatic lobule. They enter the bile canaliculi, from there to the hepatic duct, and through hepatic duct they reach the gallbladder. This is the gallbladder, and this is the storage unit of the bile, and from gallbladder there is a cystic duct through which the bile secretion flows, and the cystic duct joins to the pancreatic duct and become common bile duct, and from there to the ampulla of the Vater, and then to the duodenum. So between the ampulla and the duodenum there is a sphincter called a sphincter of Oddi. Okay, so here you are able to see liver, the hepatic duct, and bile canaliculi, and with the help of hepatic duct reaches to the gallbladder, storage unit of the bile secretion, and from there to the common bile duct and opens into the duodenum, the first part of the small intestine. Let's learn about the functions of liver. Liver is considered as one of the most important internal organs because it has several functions like secretion of bile juice and bile pigments. It plays a key role in carbohydrate metabolism. That means it carries out glycogenesis, glycogenolysis, gluconeogenesis, and lipogenesis processes. What is meant by them? First, glycogenesis means conversion of glucose into the glycogen is called glycogenesis. Okay, it is done by the insulin, which is a hormone, and glycogen is a stored form of glucose in the animals. Glycogenolysis, that means whenever we require glucose, the stored glycogen converts into glucose. The process is known as glycogenolysis. Next, gluconeogenesis. It means the formation of glucose from non-glucose, non-carbohydrate substances. Okay, so fatty acids and proteins are non-carbohydrate substances. From them, if the glucose is generated, the process is known as gluconeogenesis. Lipogenesis is the formation of fat. Right. Next, the ornithine cycle takes place in the liver. It is the conversion of ammonia to urea by the process of deamination of proteins. That means amine group is eliminated from the protein structure, hence the very harmful ammonia converts into the urea. Okay? Ammonia can become the poisonous substance in the blood if it continues to flow. So whatever ammonia produced in the body, it is converted into urea. Hence urea is a little harmless substance. Okay, the process is known as ornithine cycle. And the Cori cycle also take place in the liver, in which the lactic acid convert into the glucose. So whenever we do some physical exercise or any vigorous exercise, our body carries out anaerobic respiration. That means it has no involvement of oxygen to break down the food. So in the anaerobic respiration, lactic acid is produced in the cells, and lactic acid is the reason for the muscle cramps. So when we do exercise or any vigorous activity, we find pain in the muscles, and the pain is because of this lactic acid, and eventually lactic acid converts into the glucose, and we'll get the relief from the pains. And this process is called as Cori cycle and takes place in the liver. Next, it detoxify the substances. Okay, whatever the absorbed food nutrients are there from the small intestine, they enter into the liver with the help of vessels, and they are detoxified and supplied to all parts of the body. That means poisonous substances are removed. And next, it is a hemopoietic organ in fetus and erythroclastic in adults. Means in the fetus, that means in the embryonic stage, it produces the blood cells, hence it is called hemopoietic organ, right? And next, it acts as erythroclastic organ in the adults. That means breaking down the RBC. Okay, in the fetal stage, it produces the RBC, and in the adults, it works as a erythroclastic organ. Actual erythroclastic organ is spleen, but of liver also helps in this process. Next, synthesis of plasma proteins like albumins and globulins, and they also synthesize anticoagulants like heparin, which is a natural anticoagulant. That means it doesn't allow the blood to clot inside the blood vessels. Okay, and it also produces the clotting factors like fibrinogen and prothrombin. Okay, it synthesizes several substances like albumin, globulin, anticoagulant called heparin, clotting factors like fibrinogen and prothrombin. So liver contains the Kupffer cells, which are actual macrophages. Okay, so they are present at the sinusoids, and they kill the pathogens which enter into the liver. These are the several functions of the liver. Let's learn further. Let's learn about the structure of pancreas gland. Pancreas is the second largest gland in the body, and it is also called as a mixed gland because it has two parts, the endocrine part and the exocrine part. There is a difference between endocrine glands and exocrine glands. Basically, endocrine glands releases their hormones directly into the bloodstream, whereas exocrine glands contain a duct, and they are called duct glands. Okay? And endocrine glands are called ductless glands. So pancreas has two parts, in exocrine part which is called pancreatic acini, and endocrine part which is called islets of Langerhans. Okay, exocrine part of the pancreas, which are pancreatic acini, they produce pancreatic juice which contains several enzymes in it, and here you are able to see the lobule-like substances, and these are called as pancreatic acini. And between the pancreatic acini you are able to find island-like substances or parts. These parts are called as islets of Langerhans. Islets of Langerhans helps in the secretion of insulin and glucagon. Okay? So insulin basically converts the glucose into stored form, which is glycogen, and glycogen is stored in the liver. Okay, and glucagon helps in the conversion of glycogen into the glucose. Okay, pH of the pancreatic juice is 8.4, which is high alkaline in nature. Okay, so if the pH is 7 below, it is called acid, and if the pH is 7 above, it's called alkaline. You know about it. Okay, so in the next class we will be learning about the actual action of all these enzymes which are secreted by different kinds of glands. Okay.